Hello everyone, welcome back to our journal club meeting. Today we have Morgane, a PhD student from University of Otago, supervised by Kirsty. Morgane's paper already on the screen. Morgane, do you mind if we stop you for questions? If they
Absolutely, feel free. Yeah, I'd love it to be a bit of a discussion.
So yeah, feel free to post your questions to the chat and we will start meanwhile, or we can have a discussion after that. Welcome, Morgane, your turn.
Kia ora koutou. Thank you so much for having me here today. I am a PhD student at the University of Otago based in Wellington, and I'm part of the Wellington Cardiovascular Research Group. So we're interested in doing basic science research, translational and clinical research, looking at the pathology that leads to poor heart health. In particular, we're quite interested in improving risk stratification of patients as well. So I'm the first person in my group to delve into the EV side of the research. But fortunately, I'm in the same department as the Wellington Surgical Cancer Research Group, which is led by Dr. Kirsty Danielson. So I'm in very good company. So just before we go into the study, I just thought I'd do a brief outline of my research. So what I'm looking at the moment is human cardiac fibroblasts. And we're really interested in looking at how they coordinate repair following a heart attack. So a lot of cell-to-cell communication goes on. So that's why we're quite interested in delving into the extracellular vesicle world. And in particular, we're looking at how we can measure EV output in cardiac fibroblast in a reparative environment for biomarker discovery, as well as understanding more detailed crosstalk between the cells. In particular, I'm quite interested in doing RNA sequencing on these exosomes or EVs as well. And that's also why I'm quite interested in the paper that we're going to discuss today. And the image on the screen is some of the cardiac fibroblasts I'm growing at the moment in culture. So the study that we're going to discuss today is exosomal link RNA AK139128, which is a bit of a mouthful, derived from hypoxic cardiomyocytes, promotes apoptosis and inhibits cell proliferation in cardiac fibroblasts. This study was published about a month ago in the International Journal of Nanomedicine. Was conducted by Wang et al., which are a group from China. And the reason that I chose this paper is because it uses a lot of the experiments that I plan to undertake, and I was very interested to see how they navigated that, but also how they published that in their paper. So a little bit of background. This group is interested in the disease model of myocardial infarction, which is commonly known as a heart attack. This is a significant health and economic burden worldwide and is the acute form of coronary artery disease. So the pathology of a heart attack occurs early in life, and it starts with a chronic inflammatory disease known as atherosclerosis, which leads to plaque buildup and cholesterol in the coronary arteries. When this leads to occlusion, this impedes the blood flow to the heart, and this can lead to oxygen deprivation of localized cardiac tissue. And this process is known as hypoxia. And prolonged hypoxia can lead to mass cell death, such as through mechanisms of apoptosis and necrosis. And this is the sort of pathology this group is interested in investigating further. So cardiomyocytes and cardiac fibroblasts are two key cellular components of the heart. They're the most abundant, and they're also critical for the repair process following a heart attack. So cardiomyocytes are heavily affected by hypoxia, and they are the cell that has a large cell death. And this is a significant problem because they have negligible regeneration capacity in adults. So once they have died, they don't regenerate. So that's where the cardiac fibroblast comes in, and that cell is important for filling in the space where those cardiomyocytes once occupied. So recent studies have indicated that extracellular vesicles and long non-coding RNAs can propagate hypoxia-induced apoptosis. And understanding this process in more detail could help us understand not only the pathology of the disease, but potentially harness EVs as therapeutic targets for repair. And in particular, a new link RNA, AK139128, has been shown to promote cardiomyocyte apoptosis and hypoxia. So this leads to the aim of this research group, in which they wanted to examine how hypoxia-induced exosomes from cardiomyocytes could modulate cardiac fibroblast activity. And they had a specific emphasis on link RNA AK139128. So before we dive into the figures, I thought we'd just go over the basic methodology that they use continuously throughout this study. So the cardiomyocytes were isolated from the ventricles of 3 adult rats. These cardiomyocytes were grown in culture, and then 1 million cells were plated out and stimulated under normoxic or hypoxic conditions. So normoxic conditions were not specified exactly in the study, but we will assume it's under the standard 5% CO2 and 37 degrees Celsius incubation. So hypoxic conditions were achieved through 2 processes. The first was administering the cells with a low glucose and anaerobic DMEM that had been pre-treated with 95% nitrogen and 5% CO2 for a minimum of 20 minutes. These cells were then treated under hypoxic conditions, which were defined by 1% oxygen for between 48 and 72 hours. I couldn't find an exact precise time that they used for all of their studies following on from this, but we'll just assume that's a standardized time they decided worked best. From here, once the cells had been incubated under the conditions, cell culture supernatant was isolated and cell debris was removed through a 10,000 G spin for 30 minutes and filtration. The purified exosomes were isolated from the cleared supernatant using differential centrifugation. So we had 120,000 G spin for 2 hours to pellet the exosomes, and then they had a PBS wash and they resuspended the purified exosomes in 500 microliters of PBS. So some of these exosomes were then taken through to the cardiac fibroblast co-culture. And the way the study describes their standardizing of this process was taking some of the exosomes and using a BCA assay to measure the protein concentration. And from here, they identified 1 microgram to 5 micrograms of exosomes, which were used in the co-cultures, which occurred for 48 hours. And it's unclear from methodology if the cardiac fibroblasts that were also isolated from heart cells of rats were from the same rat as the cardiomyocytes or different rats. All right, so through to figure number 1, which was interested in looking at the purity of the cell cultures and the characterization of the exosomes. So figure 1A used Western blot analysis to confirm the presence of cell-specific markers for the cardiomyocytes and cardiac fibroblast. At this stage, it's important to note that there are no one cell-specific marker for either of these cells. Instead, a combination of markers are used to identify them, which we can see that happening here. And that's why you'll notice in the Western blots that it appears all of the markers are expressed by all of the cells, but just in lower levels. And figure 1B is just a phase contrast microscopy image of these cells in culture. So 1C uses, in the images we see here, transmission electron microscopy for normoxic and hypoxic exosomes from the cardiomyocytes. And in these images, we can see the lipid bilayer membrane particles visualized. And then nanoparticle tracking analysis was used to demonstrate particle size. And we can see that within normoxic and hypoxic conditions, EVs or exosomes are between 100 and 200 nanometers in diameter. And figure 1D uses a Western blot analysis to demonstrate the presence of the EV-specific markers for normoxic and hypoxic conditions. So overall, I think this figure is quite good. I think there's a few things that could have been included to improve it a little bit. For example, in figure 1D, I think it is important to include our cell-specific marker that's negative in exosomes such as calnexin. I also think it's quite important when you're looking at 2 different stimulation groups, particularly one that you know can lead to cell death, to include these statistics or cell viability between the 2 groups, because obviously cell death is going to increase the number of exosomes. And on that note, we don't have, even though they've used particle tracking analysis for size. They haven't shown the concentration. I wonder if this is because this isn't necessarily the novel part of their study. The novel part comes a little bit later on, so perhaps they felt like this had already been done a lot in the literature. But I think that using the concentration from the NTA would have been useful for standardizing their EV concentrations at this stage, and then maybe using the BCA assay to complement this as well, because they do use co-culture of their EVs throughout the rest of the experiment. Does anybody have any points to add to that?
Morgan, yeah, there is a question in the chat about one of the figures. So Olivia asked, if the figure B images look very similar, do you expect or authors to see the difference in morphology?
Yeah, so they do look quite similar. I haven't actually worked with cardiomyocytes, but a lot of the typical structure of the cardiac fibroblast is the spindle shape, whereas cardiomyocytes more have a sort of myocyte morphology. However, I don't think that a phase contrast microscopy is enough to visualize this. I think that if they were really wanting to look at the physical aspects of them, they should also use confocal microscopy, which can show that those actin filaments. But I think for the purpose of this study, what they've provided is enough.
Mm-hmm. Thank you.
Cool. Alrighty, so now that we see that cardiomyocytes produce, also actually I should note at this point, I'm going to continue using exosomes because that's what the study discusses, but EVs probably is the more appropriate term to use. But so now that we've established that cardiomyocytes produce exosomes under normoxic and hypoxic conditions, the next study was to look at how these exosomes influence cardiac fibroblast activity. So figure 2A uses a CCK8 assay to look at cell proliferation of cardiac fibroblasts. And what we can see in this figure on the y-axis is the optical density value, with higher values meaning more cells are present. And on the x-axis is the time at which these measurements were taken. So 3 groups were examined. We have our control group, which is PBS co-incubation, and then our normoxic exosomes and our hypoxic exosomes. And we can see from this proliferation assay that co-incubation of cardiac fibroblasts with exosomes decreases their proliferation. And this is most significant in hypoxic exosomes. So the next test they did was to look at migration and invasion of cardiac fibroblasts in the same 3 culture groups. So the migration and invasion assays were done in a similar manner. They used a transwell plate. So this is when the cardiac fibroblast is seated in an insert and the test media is at the bottom of the plate. And between the cells and the media is either a cell permeable membrane for migration or a matrigel for invasion. And at this point, it's important to say that these 2 parts of cellular activity are quite important for cardiac fibroblasts, particularly in repair, because migration, we want to see the cardiac fibroblasts moving to repair. And invasion, our cardiac fibroblasts are the main source of extracellular matrix in the heart. So their ability to synthesize and break down extracellular matrix is really important. So when the cells move through, after incubation, the cells move through the gels, and then the cells are stained and counted. So the images are found in figure E and F for migration and invasion respectively, and quantification of this is shown in B and C, with the number of migrated cells on the y-axis and our conditioned groups on the x-axis. And what we can see is similar to proliferation, when cardiac fibroblasts are when they're co-incubated with exosomes, they have decreased migration and invasion activity, and this is most pronounced in our hypoxic exosomes. So figure 2 also looked at cell apoptosis, and they used a tunnel assay which detects DNA fragments that are formed during apoptosis. And in figure G, we can see staining for the tunnel product in the first column, and in the second column we have DAPI for staining the cells, and then we have the merged on the end. And through the quantification, we can see that there are more apoptotic cells in the hypoxic exosome co-incubation than the control PBS and the normoxic exosomes. So what we can see from this figure is that the cardiac fibroblast activity is significantly inhibited by our hypoxic exosomes, and they also have increased apoptosis when they are co-cultured with them. It's important also to note with these studies and throughout the rest of the experiments, they don't necessarily say how many replicates they've done of each study, which is something I think they should be including. But yes, that's that figure. Does anybody have any thoughts about that? No, cool. Okay, we can move on. So now that we know that incubating cardiac fibroblasts with these exosomes leads to decreased activity, this group wanted to investigate how this could be happening. So figure 3 looks at the uptake of exosomal RNA into cardiac fibroblasts. So the exosomal RNA was fluorescently labeled with ExoGlow, which is EV specific labeling kit, and was imaged. And we can see just based on this imaging that the exosomal RNA is uptaken by the cardiac fibroblasts, which suggests that perhaps a mechanism of action is that the exosomal RNA is transferred to the cardiac fibroblast RNA, which could be affecting its activity. Again, with these images, just looking at them, it appears that there's more RNA in the hypoxic exosome group than the normoxic exosome group, which again could go back to discussing how hypoxic exosomes do have, there may be more hypoxic exosomes, so it goes back to the idea that perhaps they should have been measuring the concentration and standardizing it using NTA. Cool. Alrighty, so following on from this, the group wanted to investigate what the RNA looked like in the exosomes. So figure 4 is quite a hefty one, but it's to do with our RNA sequencing. So total RNA was isolated from the exosomes, and RNA libraries were constructed using TrueSeq standard total RNA library prep. And then the RNA sequencing was performed on Illumina HiSeq 2500. So candidate link RNAs were identified between the two groups if they had a twofold change with an adjusted p-value of less than 0.05. And qRT-PCR was used to validate the top 10 upregulated and downregulated link RNAs. So figure 1A is just our volcano plot of the RNA sequencing to show our RNAs that were upregulated and downregulated. And figure 4B is the top 10 differentially expressed link RNAs between the normoxic and the hypoxic exosomes. And figure 4C is the heat map, which demonstrates the validation of the qRT-PCR as it follows the similar downregulation, upregulation between the two groups. Just before we move on from this, there's a few, because I'm quite interested in this area, because this is what I'd like to do, there's just a few things that I found quite interesting. So first of all, they didn't include a lot of validation, discussing how they validated their RNA sequencing, which I would have thought to expect in something like this, but at the same time, this paper's not entirely focusing on RNA sequencing, so that may be why they didn't include that information. Also, it's with our qRT-PCR validation, they didn't say how many samples they checked this in, and they didn't explain if this was a biological replicate or technical replicate, which I think would have been nice to see as well. So from this information, they took the link RNA AK139128, and their rationale for choosing this RNA over all the others was that it was just one of the most upregulated RNAs in the hypoxic exosomes, which seems to be quite common within papers. I'm not sure if this group has a pre-interest in this RNA or if it was one they selected based on the literature. Yes. So from here, they then used qRT-PCR to measure the levels of link AK139128 in a number of different experiments. And the qRT-PCR housekeeper was beta-actin. So in figure 4D, we can see that under normoxic and hypoxic conditions, the levels of AK139128, which I think I might just refer to as AK1 from here on out, is higher in the hypoxic group, which further validates their RNA sequencing. So next, to further validate their sort of findings, they took the exosomes and treated one group with RNAs and an RNase inhibitor with or without Triton X100, which is used to lyse the cells.
Morgane, sorry.
Oh yes, hello, yes.
Sorry, on that last one,
yes.
On the last slide where you've got the list of genes, I'm just like looking at that list going, HIF1A antisense 1.
Yes.
So the antisense long non-coding RNA to HIF1 alpha wasn't interesting to them for hypoxia.
So this is what I found quite interesting with this as well. And if you've done RNA sequencing before, particularly with non-coding RNAs, I'd be really interested to hear. But I feel like what I would have liked to see as well is the proportion of sort of long non-coding RNAs to the mRNA they also got from this, because it doesn't seem to be that they enriched for long non-coding RNAs, and they've sort of just selected a really small group. But like you said as well, within their RNAs, they seem to have just picked an arbitrary link instead of link RNA, instead of picking some other ones that also look really interesting. So I'm not sure if they've also tested the other links as well, and they found that this was the most interesting, or if their group had a pre-interest in this.
Yeah. It's a shame, isn't it? Because if they had tested the other ones and they were negative, it would have been nice to see that data, because someone else could go and chase it. And and I'm just looking at the p-value. The p-value is the best for HIF1A antisense.
Yeah.
And that means that this AK, and we're really liking the catchy name, AK139128.
Yeah. Yeah. No.
Number of a Russian machine gun.
Yeah, I know. I've got to be. Yes, no, I completely agree with that. No, and it's really interesting as well, because they've picked this one, and they've said they've picked because it's one of the most upregulated. But if you look at the log square values, they're not much different, are they, between the top?
No, and they're very, very tight. I'm actually surprised to see so many with the similar fold change at 8. So that makes me wonder about their depth, and that was one of the questions.
Yes. Oh, perfect. Yes.
About the depth of the data.
Yes.
How many reads did they actually have? It is a bit suspicious.
Yes, that is another great point. No, they don't have that information in their RNA sequencing of the depth, and I think that could be a problem as well, because if they haven't enriched for long non-coding RNAs, then I feel like their sequencing was probably swamped by mRNA. So that could also be a massive problem. Yeah, no, it is interesting that they didn't look at anything further, and they haven't explained that at all either. So maybe they've got some more papers coming out looking at that. I'm not sure. But yes, no, onwards with AK139128. So what they found when they used RNAs and RNase inhibitor with Triton X100, with or without, they found that when Triton X100 was incubated, the levels of AK139128 were decreased in both normoxic and hypoxic exosomes, which suggests that this RNA is found within the exosomes. And within this figure as well, they wanted to look at potentially the potential transfer of link AK139128 between normoxic exosomes and hypoxic exosomes. The transfer of it from the normoxic exosomes and hypoxic exosomes to the cardiac fibroblasts. So when they incubated different doses, they found sort of a dose-dependent increase in the levels of this link RNA for the cardiac fibroblasts, which they suggest could be associated with the change in cardiac fibroblast activity when incubated with these exosomes. And I haven't missed figure 4E. I left it to the end because I think it flows quite nicely into their next set of experiments, in which they transfected cardiomyocytes with a control or AK139128 short hairpin RNA plasmid to knock down the expression of this link RNA. And this quite nicely shows that in the presence of the inhibitor in cardiomyocytes and subsequent exosomes, the expression of this link RNA is downregulated. Yes, so from here, they wanted to investigate, now that they know that this candidate link RNA is expressed in hypoxic exosomes and potentially could be important, they wanted to investigate what would happen if they incubated these hypoxic exosomes with the cardiac fibroblasts when this gene is downregulated. So that is what this next figure looks at. And this is the last of our in vitro experimental work as well. So we're back to the proliferation assay, which is again is CCK8, and our groups this time are control, which is PBS incubation, and hypoxic exosomes, which either contain the control inhibitor or the link RNA inhibitor within the exosomes. And what we can see is, again, similar to the first assay, when our hypoxic exosomes are incubated with cardiac fibroblasts, there is a decrease in proliferation. However, when this link RNA is inhibited, the proliferation rates go back to the control levels, suggesting that the inhibitory effect is abolished when you inhibit AK-139-128. And this is really similar with the rest of the activity studies for cardiac fibroblasts. So we can see when the hypoxic exosomes are co-cultured with the cardiac fibroblasts, when AK-139-128 is downregulated, the levels go back to the control. And we see this again with our tunnel assay. So cardiac fibroblast-induced apoptosis by hypoxic exosomes is abolished when this link RNA is inhibited. So I think that these assays look very convincing, almost too perfect, to show that this link RNA obviously has quite a significant effect on the cardiac fibroblasts. So from this in vitro work, the group then wanted to do some in vivo studies. So the methodology that they used was they took the rat model and they gave it an artificial heart attack using left anterior descending coronary ligation, which is essentially blocking the LAD artery in the heart. From here, they then took either a PBS control or hypoxic exosomes, either with the control treated exosomes or the AK inhibited exosomes. And they placed this in 4 remote regions around the dead tissue site. So they left the rat for 3 weeks and then it was sacrificed and the cardiac tissue and cells were analysed. So figure 6A demonstrates that there's an increased concentration of AK-139 in cardiac tissue and cardiac fibroblasts that have been treated with the control exosomes compared to the exosomes that have this link RNA inhibited, which suggests that, that there's some sort of transfer occurring to the tissue and to the cells. So from here, they again do their migration and invasion assays just with the cardiac fibroblast cells isolated from the rats. And they found that rats treated with the AK-139-128 exosomes had improved invasion activity and migration compared to the control treated rats. So again, it's showing that when this link RNA is downregulated, the inhibitory effects on activity are abolished. And figure 7 looks at apoptosis using a few different methods. So again, the figure 7A and B is using our tunnel assay, and they show very similar results to what we saw in the last set, in which apoptosis increases in cardiac fibroblasts that are treated with hypoxic exosomes with control inhibitor, and this effect is abolished when the link RNA is downregulated. And they follow this up with figure C and D, which they measure BCL2 and BAX protein. And I just have some notes on this here. So the BAX protein is part of the BCL2 family, and it promotes cell death, whereas BCL2 prevents apoptosis by inhibiting BAX. So they show, using a Western blot analysis, that the presence of BCL2 and BAX is most strongest in our exosome groups. And when they do the ratio of this, they show that there's a slight decrease in the link inhibited exosome cardiac fibroblasts, which suggests that they may be more resistant to apoptosis, although this graph is a little bit less convincing than our tunnel assay. Yes. So overall, oh, do we have a question? No, okay. So overall, hypoxia-induced exosomes derived from cardiomyocytes significantly inhibited cardiac fibroblast activity and increased cellular apoptosis. They found that this link RNA was significantly upregulated in hypoxic exosomes compared to the normoxic exosomes, and when this link RNA was inhibited in the hypoxic exosomes, its inhibitory activity was abolished in cardiac fibroblasts in vitro and in vivo. I think that this paper had a nice logical set of experiments that they followed through, which I found quite interesting because a lot of it was done in vitro, and a lot of the stuff I will be using as well. I do think they could have used some more information in their methodology. I think that that's what's created a little bit of confusion, and I think that with the RNA sequencing, if they were particularly interested in one type of link RNA, that perhaps RNA sequencing wasn't the best approach and they could have used a different gene expression analysis methodology. But overall, I did really enjoy this paper, and for me, I found it quite interesting. Yeah, do people have any final thoughts about this?
Thank you, Morgan. We have a comment from Olivia about figure 6E.
Oh, yes.
So the comment is, figure 6E, the invasion assay don't look very even. So how do you know, or authors, that the field of view was representative of the whole assay? The controls also look very weakly, fluorescent.
Is this for this one here?
Oh yeah, maybe.
Yeah, this might be here. Yes, very good question. You can see here that the exposure seems increased compared to these other two. Yes, that's a very good point. If it's actually what it is. Do any other people have opinions on that?
I'm finding a lot of the graphs suspicious. If you have a look at the graphs when you go back, the error bars are really tight, and the recovery, that recovery back to being perfect by knocking down
one link RNA.
I have never seen that before.
Yes.
And
so like I said, the thing that's quite frustrating with this paper is they don't say how many replicates they've done for each of them, which I think that they absolutely should have. But you're right, it's standard deviation, and it's very, very small.
It's very small, and I've never seen any that I can't
recover so well. Yeah, that's why.
Perfect. It is just I'm feeling that I'm looking at these invasion assays and trying to look for like dodgy repeated areas and just seeing if this is a manufactured paper, because vicious I'm feeling.
So this is a good question. So if people were to publish work that's a bit suspicious, is this sort of the area that the peer reviewers should sort of find out, or how does that process work?
Yeah, I mean, it depends on the peer reviewer. It depends what they're looking for. If you've got lacking methods, then if I was reviewing it, I would go back and I would ask them for it and tell them, I would say, I want to know your sequencing depth. I want to know the data so that I can understand that this is good.
Yeah.
It seems to be a very extensive paper, and I don't really know the journal very well. It seems to be it's not a very well-known journal, and I in around the houses. I can't remember the journal.
Oh, the International Journal of Nanomedicine.
The International Journal of Nanomedicine, that popular tome. Do you feel suspicious about it? How does Kirsty feel? Kirsty, how do you feel about it?
Kirsty's not here.
Oh.
Those are
Yes.
investigations that are incredibly tight error bars.
Yeah, they've got a robot working there.
I mean, one thing that would be nice to see with these plots is, and a lot of journals are insisting on this now, is that you don't show bar graphs anymore.
Oh
Yeah.
So you have scatter plots.
Yeah.
Yeah.
Yeah, I'm a big fan of that. Single data points so that you can see the variation. You don't know if they've got like two distributions in them. And also the cut out Westerns, I don't know if, they've got them.
Yeah.
Mm-hmm.
We're just being nitpicky, but
No, but again, there's rules about how you present Westerns. I just
Yeah.
But you're actually supposed to show a full-length Western.
Show what? A full-length Western.
yeah, instead of a trimmed one, having it supplementary. But then some journals don't allow you to have supplementary, which is crazy. But I'm trying to find patterns. I don't know if any of you on Twitter follow Elizabeth Bick, but she pulls up copy pasted papers where she looks for, it's really good. You should follow her. It's really, really interesting. But yeah, I'm now trying to see if I can see patterns, because I'm just feeling a bit suspicious about those bar graphs.
Wow. So is there enough papers out there that this happens quite a lot, that there's a Twitter account dedicated to it? Oh, wow.
There was a recent one where it was, I can't remember, 8 papers, and they were all on different topics, but they all came from different groups, but all the figures were the same.
Wow.
What? That's so weird.
I don't mean this might be a very nice paper, and might be, but I'm just those bar graphs,
It is. The other thing about this as well is that I think this is the clinical application of this is quite significant. If you can show that you've got this one RNA that can affect cardiac fibroblast activity. They didn't really sort of scream that from the top of the hills in their discussion, and I think that that's a really important aspect of the study as well. Have been emphasised in their discussion a little bit more. Yeah.
No, interesting. Very interesting.
Mm.
I mean, the approach for the methods is good. I mean,
Did you want to say anything?
Thank you, Morgan. Do we have any other questions? No more questions. Nothing in the chat. Thank you, Morgan.
Cool. Thank you very much.
yeah, that was awesome. Thank you.
Thank you very much.
Just a reminder, we don't have a meeting next week. We will meet in 2 weeks.
We're hoping, we just need to get this confirmed, but we're hoping to have a special guest who's going to present his research from Melbourne. Just to mix things up a little bit. So we will keep you posted once we get any confirmation email from him, but it'll be a good one, I promise.
Awesome.
Tell him off if it isn't. But if anyone's also got any ideas about approaching international Australia, then just let us know. We can contact people, and if there's anyone you really want to invite or give a talk, then we can approach them and bring them on virtually as well.
National speakers are very welcome.
Oh yeah.
As usual. So guys, if you want to present a paper of choice or your own research results, please contact me, and I will be glad to hear from you about that. Thank you everyone for joining us today. We will see you back in 2 weeks. The video will be on the YouTube shortly. Thank you.
Thank you.
Bye.
Thanks.
Bye.
Bye.
Oh, I just like